guys, welcome to our channel. This is Science Lectures 256. Thanks for being with us. Please consider subscribing, comment in the comment section, and like our video. So we are still continuing with our series of Monera, where we have bacteria, and we are looking at bacteria. Today we are going to look at classification of bacteria, the different methods we can use to classify these bacteria. So we are going to start with classification by shape. We are going to start with shape. So we are classifying basing on shape of the bacteria. And the first shape we are having is what we refer to as bacillus. Bacillus. The bacillus or rod shaped. So these bacteria are rod shaped. So they are shaped in form of a rod. For example, so we can have different bacteria that are rod shaped. For example, examples of such bacteria, we can have bacteria like Bacillus anthracis, and this one is the bacteria that causes anthrax. Is a bacteria that causes anthrax. This Bacillus anthracis. If you were to examine it, observe it under a microscope, you will see it rod shaped. It is shaped in form of a rod. So that gives it a shape, the word bacillus, being bacillus. So then maybe another example of a bacteria we can give to expound more is E. coli. E. coli. E. coli is also rod shaped. So if we are to have the basic drawing of our E. coli will be, then it will have numerous flagella surrounding it. And this is having numerous flagella surrounding it. This is what we called pelitrichus. And this was in our video when we were introducing the bacterial cell. If you didn't see the video, please go check it out. We said pelitrichus, where we have many flagella surrounding that. Then we said the monotrichus for one flagellum. Then we said amphitrichus, where we have flagella on both sides. So that is the rod shape. Most rod shaped bacteria have a flagella. Most rod shaped bacteria will possess the flagella. Examples, we've given two basic examples: Bacillus anthracis that causes anthrax. Then another example is the E. coli. E. coli is very, very important within the ecosystem, within our ecosystem, because E. coli we will find it in the gut of humans. In the human gut, E. coli is present, and in the human gut, it synthesizes vitamin K and B vitamins and vitamin B, which are very essential for our survival. However, the same E. coli can be used in insulin production. In the production of insulin, synthesis of insulin, E. coli can be used. Then again, E. coli is an agent of disease, can cause diseases like some urinary tract infections can be caused by E. coli. Then the same E. coli can also cause some other deadly diseases among humans, the enteric. Then we can also use the same E. coli to detect contamination of water with fecal matter. So water that is highly contaminated with fecal matter will contain these E. coli. However, the point is a classification by shape, and we are having bacillus that are rod shaped. For example, Bacillus anthracis and the E. coli bacteria. So that is it for the bacilli shaped bacteria. Then we have the bacteria that are either spherical. So we are done with the rod shaped, which is bacillus or rod shaped. Then we have other bacteria that are spherical. They appear to be spherical. When you observe in the microscope, you will see them as being spherical. These spherical bacteria are referred to as, this shape is referred to as cocci. So they are cocci. Whenever you see a spherical bacteria, it is referred to as cocci. If it has one sphere, one sphere, one circle, we can call it monococci. Monococci. We call it monococci, being monococci. Then if it has two, we refer to it as diplococci, diplococci, or diplococcus. Then if there are many, a cluster. If we have these ones are two spheres, then if we have a cluster of bacteria, a cluster that is shaped like grapes. In fact, this is you're like as if you're seeing grapes. This is what we refer to as staphylococcus, where we have a cluster of these bacteria is staphylococci. For example, which bacteria can we give as an example here? Is staphylococcus, a bacteria known as Staphylococcus aureus. Staphylococcus aureus, this is a bacteria that is will appear as clusters. And this bacteria is a normal flora. Is the normal flora, found as a normal flora on the skin of humans. However, it can result into skin infections once it becomes an opportunistic pathogen. Skin infection. So this one is staphylococci because it is shaped. We have clusters of the cocci. If we have only one cocci, that is monococci, two cocci, that is diplococci. Then if we have another presentation, like if we have chains, these are now chains of the cocci. This is what we refer to as streptococci. So in strepto we have a chain of bacteria of the cocci joined streptococci. The basic example of a bacteria we can give here is Streptococcus. Streptococcus bacteria that causes strep throat in humans. So that is how the Streptococcus will appear. Then if we have a rod that is curved, we have a curved rod. Rod. This is what we refer to as vibrio. The shape is vibrio. For example, Vibrio cholerae. The one that causes cholera is a vibrio shaped, Vibrio cholerae. Then we can have also another vibrio that is long, like longer than Vibrio cholerae. This is what we call, such a vibrio is what we call a spirochete. And an example of this of such a bacteria is Treponema pallidum. Treponema pallidum, the one that causes syphilis. Treponema pallidum, which is capable of causing syphilis in humans. So in summary, this is classification by shape, and we are saying we can have bacteria that are rod-shaped, and we refer to them as bacillus. For example, Bacillus anthracis, the one that causes anthrax, even E. coli. But however, we say the rod-shaped bacteria or bacilli will always, most of them have flagella, but it depends on the location of where the flagella is. Then we say for spherical shaped, we give what we call them bicocci, and if it is one sphere, we call it monococci. For two spheres, they will be called diplococci. Then if it's a cluster, a bunch of the cocci or a grape-like structure, that is what we refer to as staphylococci. For example, Staph aureus, which is a normal flora on the skin of humans, or is found on the skin of humans. However, it causes skin infections if disturbed. Then we can also have a chain of the cocci, which we refer to as streptococci, like the strep throat bacteria. Then we have a curved rod, a rod that has a curve. That is what we refer to as vibrio, like in Vibrio cholerae. Then we can have also a long rod, which is curved entirely, which is wavy, and that is what we refer to as spirochete, for example, in Treponema pallidum, the one that causes syphilis. So that is our basic classification, basing on shape. Basing on the requirements. Basing on certain requirements, specific requirements. And here we are going to put our emphasis on the requirement, which is oxygen. We shall have our emphasis put on oxygen. So oxygen, bacteria can be classified based on the requirements, and we are going to look at oxygen. So if we are to look at oxygen, we know that some bacteria are either aerobic. Aerobic, that means they require oxygen for their survival. Anaerobic, such bacteria don't require oxygen for their survival. However, some bacteria are obligate anaerobes. Then some other bacteria will be referred to as facultative anaerobes. And the same applies to aerobes. We can have obligate aerobes and facultative aerobes. So what does it mean? We say aerobic, such bacteria will require oxygen for their survival. Then anaerobic, no oxygen, but they will still survive, no oxygen present. Such bacteria will survive. But now we have obligate and facultative. So we need to know what obligate and facultative mean. If a bacteria is an obligate anaerobe, that means it requires only and only anaerobic conditions. Because it is an obligate anaerobe. It will require only anaerobic condition. For example, methanogens, bacteria that are methanogens, like Methanococcus, specifically Methanococcus, and Methanospirillum. They are obligate anaerobes. That means they live in regions where there is typically no oxygen, totally no oxygen, in those oxygen-depleted areas. So which example of a region where they live can we give? We can give the very deep layers, or the bottom of the seas. We'll find these Methanococcus. However, they are very important. Methanogens are very important because they will break down the organic matter to natural gas, or what we call biogas, methane, which can be used for other important functions. And they can also be used in bioremediation, and this involves cleaning of our environment by breaking down that organic matter and changing it into different other components that might be very important for our environment and for other uses. So that is what we mean by being obligate anaerobe. Anaerobically obligate, that means they are typically don't require any form of oxygen to survive. They require oxygen-depleted environments for their survival. Methanogens credited for that. Then facultative anaerobes. So, for example, so these facultative anaerobes will live in anaerobic areas. In other words, they can survive in anaerobic areas. But however, they can also survive in presence of limited oxygen. In presence of limited oxygen, they can survive. For example, the bacteria that reside within our guts, within the guts of warm-blooded animals, warm-blooded animals like man. Bacteria which reside there are called E. coli. They are facultative anaerobes. Because they live in your gut. That means that the gut has limited oxygen. Limited oxygen, however, the gut is not that oxygenated. So such bacteria, the enterobacteria, the ones that live in our guts, are facultative anaerobic bacteria. Then we have also obligate aerobes. These ones require obligate aerobes. That means they survive only and only in presence of oxygen because they are obligate aerobes. And we have examples of such bacteria. The number one example we can give here is Nitrobacter. Nitrobacter requires oxygen for its survival. So minus the oxygen being present, this Nitrobacter can die off because it is lacking oxygen. It is an obligate anaerobe. And we know Nitrobacter is very important in converting the nitrites to nitrates that will be utilized by plants for their growth. Then we also have bacteria that are facultative aerobes. Such bacteria are aerobic, but as well they can survive in presence of some carbon dioxide. However, there is also another important group of bacteria that are obligate anaerobes, and those are the Clostridium species of bacteria, like Clostridium difficile, then Clostridium perfringens, then Clostridium botulinum, also that one can be an obligate anaerobe. So that is it with classification based on requirements, but the requirement we are putting emphasis on is oxygen. Some can be obligate anaerobes, facultative anaerobes. Then we can have obligate aerobes and facultative aerobes. So that is classification based on requirement. So bacteria basing on the chemical composition. So chemical composition will basically we'll have a closer look at their cell walls. So we know that cell walls of bacteria are made up of a peptidoglycan. So it's a peptidoglycan making up the cell wall. We have some peptides, those amino acids, then some glucose making up this cell wall. However, we say bacteria either gram positive or gram negatives. But the gram positives have a thick peptidoglycan layer, and gram negatives have a thin peptidoglycan layer. So the difference in such a composition brings about a difference in chemical composition. Okay, this change in chemical composition gives a different taste when we stain this bacteria with a stain like Gram's stain. So when we apply Gram's stain, gram-positive bacteria will get a purple color. We'll observe the purple color present. Then for the gram-negative bacteria, we'll observe the red color of the stain. And we will go into deeper details in our video of staining, of gram staining, how we stain these bacteria. We'll go into deeper details. So chemical composition, basically we are looking at how these bacteria behave when stained with a stain like gram stain. So when we stain with gram stain, these cell walls, gram positive, will be purple, gram negative, and the cell wall will show a red color. Then, which other classification can we put across? A classification of enzymatic action. So different bacteria have different enzymatic action. What does this mean? This enzymatic action, we look at it basing on what is produced, what bacteria are capable of producing, what is produced, and what they consume or degrade and how they degrade it. But we look at what bacteria produce. Certain bacteria, and how they degrade different substances, basing on the enzymatic action. Which enzymes do they have, and how do these enzymes do their roles, and what role do they do? For example, we know that some bacteria will convert nitrates to nitrates. Some bacteria can convert nitrates to nitrates, like Nitrobacter. Nitrobacter will convert nitrates to nitrates using its enzymatic action. However, some bacteria can also yield hydrogen sulfide or produce hydrogen sulfide gas. For example, we can give an example of a bacteria called Edwardsiella tada, abbreviated ETADA. Such a bacteria, one of its ways, if we do biochemical testing or biochemical analysis of such a bacteria, it will yield hydrogen sulfide gas. So we'll test hydrogen sulfide coming out of this bacteria. So enzymatic action, what the bacteria are capable of breaking down and what they are capable of producing as products can also be used in classifying different bacteria. Because we can have those that can produce hydrogen sulfide, we can have those that can convert one inorganic compound into another inorganic compound. So we can also classify bacteria in that way. Chemical composition, enzymatic action, we don't forget the shape. We also had the shape. We also had basing on the requirements like oxygen. Then chemical composition and enzymatic action we are done. Then we can also classify basing on the temperature. We very well know that all bacteria have what we call an optimum temperature. So we can have three types of temperatures here: a minimum temperature, the optimum temperature, and the maximum temperature. Minimum temperature is the temperature below which that bacteria can no longer survive. Minimum temperature below which it can no longer survive. Maximum is the temperature above which bacteria can no longer survive. Then optimum is the temperature where we'll have rapid growth of such a bacteria. So the bacteria is thriving at the optimum. So we expect rapid growth and at times rapid metabolism, of course, that leads to the rapid growth, the increased growth. However, each bacteria, we say it has an optimum temperature. But most pathogenic bacteria will have optimums at around 37 degrees Celsius, because that is at least the temperature, the body temperature for most of the humans, for most of the warm-blooded animals. So pathogenic bacteria will use that advantage of around 37 degrees Celsius to invade their hosts. But we know that we have a minimum temperature, optimum temperature, then a maximum temperature. And all these form what we call the cardinal temperature. Cardinal temperature. However, some bacteria can also survive in very low temperatures. Some bacteria prefer environments with very low temperatures, and such bacteria, so we can find such bacteria in the Antarctic and the Arctic regions. And such bacteria are referred to as cyclophiles. Then some other bacteria will survive in extremes of high temperatures. And such bacteria are called thermophiles. So this is classifying basing on the temperature. Different bacteria will have a different optimal temperature through which they will thrive. Then we can also have another classification basing on what we call serological testing. Serological testing or serology, basing on serology or serological testing. So in serological testing, what we are looking at is basically antigen. We are looking at antigen-antibody interactions. Antigen-antibody interactions. So different bacteria are designed to interact with different antibodies in different ways. So in serological testing, we have a bacteria. We'll have a bacteria tested against an antibody, because the bacteria now is the antigen tested. So we have a bacteria which is an antigen tested against a specific antibody. So this can also be used to classify the bacteria. So they will manufacture, will have a specific antibody, and we test it against the antigen, which is a bacteria, and we see the test results that will come out finally. So we can use that to classify bacteria. Then we can classify also basing on pathogenicity, the ability of the bacteria to cause disease, or to become a pathogen. We can have bacteria that do not cause any disease, and we have bacteria that can easily cause diseases. So we can classify also basing on the pathogenicity. Then we can also classify basing on nutrition, basing on the nutrition. And in nutrition, we know that some bacteria have capacity to synthesize their own food, and such bacteria are referred to as autotrophs. So these autotrophs utilize sunlight energy. They utilize sunlight energy, and they will later synthesize their own. For example, cyanobacteria. This cyanobacteria will utilize sunlight energy because it has a pigment. It has pigments. It has chlorophyll A and another pigment called phycobilin, and it will use these pigments to trap sunlight energy to synthesize food. So that makes it a photoautotroph. This bacteria photoautotroph, and in the end it will give us oxygen gas. So that is basing on nutrition. We can have bacteria that are photoautotrophs. Then we can also have bacteria that are chemoautotrophs. And have bacteria that are chemoautotrophs, and these chemoautotrophs will obtain energy from breakdown. They obtain energy from breakdown of inorganic molecules. So they obtain energy from the breakdown of inorganic molecules, and they will later synthesize food. What are the inorganic chemicals we are talking about? For example, hydrogen sulfide, carbon dioxide as the inorganic molecules that can be broken down by the purple bacteria, such that this purple bacteria obtains the energy to synthesize the food. So some bacteria can be chemoautotrophs. Then we can also have other bacteria being photoheterotrophs. So these photoheterotrophs will obtain energy, sunlight energy, to make their food. But however, they will also utilize organics or organic foods in order to. They will utilize or feed on organic forms. However, they obtain also energy from the sunlight. Those are the photoheterotrophs. For example, the basic example here we can give is the bacteria called Rhodopseudomonas. Rhodopseudomonas can be a good example of a photoheterotrophs. Finally, we can have bacteria that are heterotrophic, and most of the bacteria fall under this category, for heterotrophic. These bacteria will utilize, or they will just consume the available organic material, will utilize or consume the available organic material in order to generate energy for their survival. So that's how we can also classify bacteria, basing on the nutrition. However, we can also do some molecular testing. On the basis of, basing on DNA, basing on the genetic composition and its associated mutations, to also classify bacteria. So that one can also be used as a method of classification. Also classify these bacteria basing on their susceptibility, or how are they predisposed? Susceptibility to bacteriophages. Susceptibility is like proneness, like how are they predisposed. Susceptibility to bacteriophages. What are bacteriophages, first of all? Bacteriophages are viruses which can be used to attack or to destroy, or destroy bacteria. So these are viruses, but they can be used to destroy or attack bacteria. So in our classification of bacteria, we can classify based on how susceptible is a bacteria, is a given bacteria to this virus, the bacteriophage. So that is it with our classification of bacteria.